one of the benefits of publishing in JVS Vascular Science is the opportunity to be selected to present your work in webinar format. And as I'm sure all of you know, the webinar appears on our homepage, and all previous webinars are also in our online version, which is at www.jvsvs.org. So without further ado, I'll introduce our two associate editors, Drs. Gail Tang and Dr. Ulf Hedin, who are running today's webinar. And but before we turn it over to them, I'm going to ask Dr. Paul DiMuzio, our assistant editor and in charge of all of our audio visual, to give us the ground rules. Dr. DiMuzio.
Thanks, Alan. Privileged to help such a wonderful event once again for your journal. Just ask everyone if they don't mind to stay muted. And if you have any questions, please pose them to the chat, and our discussants will pick them up there and hopefully get to everybody's question. And once again, we are recording this, so I wish everybody a successful webinar. Thank you.
And thank you, Paul, and warm welcome to this here in Sweden late night webinar. We've had this a few times, and it's a real pleasure to discuss science. This time the webinar is dedicated to smooth muscle cells in vascular disease. And I have the privilege to introduce Professor Kathleen Martin, who is the Professor of Cardiovascular Medicine at Yale School of Medicine. She has a background in vascular science, doing a PhD at Case Western in '97, two postdocs at Harvard Medical School. And her focus has been for many years to investigate the regulation of smooth muscle cell function and how they contribute to normal vessel function and cardiovascular disease. So this is the theme of the webinar that Kathleen will now start to introduce to us. Thank you.
Well, I'd like to thank the organizers so much for organizing this webinar and for inviting me to present on an introduction to the other three talks based on a review article that we recently published in JVS Vascular Science called Targeting Smooth Muscle Cells in Vascular Disease. So we all know smooth muscle cells are really remarkable cells. So the smooth muscle cells that make up our arteries and veins, as you see here, even though they're differentiated, they retain a remarkable plasticity to de-differentiate, and this allows them to contribute to the growth, remodeling, and repair of the vasculature, but also contributes to cardiovascular diseases, including atherosclerosis, calcification, hypertension and pH, aneurysms, transplant vasculopathy, and restenosis. Restenosis and intimal hyperplasia is a complication of revascularization procedures, including angioplasty and stenting, where the smooth muscle cells de-differentiate and proliferate to participate in a wound healing response, but this response can go overboard and form these new lesions in the lumen. This is an example of these lesions in some rodent models of angioplasty or stenting. The smooth muscle cells normally reside in the medial layer where they're differentiated and quiescent, but in response to mechanical injury and the inflammatory cells and platelets and the cytokines and growth factors that they produce at the site of injury, smooth muscle cells will de-differentiate, lose their repertoire of smooth muscle contractile proteins, become proliferative, migrate into the lumen where they secrete excessive amounts of extracellular matrix that forms these intimal lesions, and they can migrate even through the struts of stents. So smooth muscle cells have this amazing plasticity to change their phenotype, not just a little bit, but quite dramatically, depending on the stimuli. And this is an example of some of these phenotypic switches that we've observed in smooth muscle cells. The one I'll be focusing on today is the one that's been best studied, is this myofibroblast transition that we see in intimal hyperplasia, restenosis, and fibrosis, where they become fibroblast-like, and we call this the synthetic phenotype because they synthesize so much matrix. But from lineage tracing and fate mapping studies, we know that they can also assume macrophage-like phenotype, and we now appreciate that smooth muscle cells contribute the majority of foam cells in atherosclerotic lesions. They can also become osteochondrocyte-like in calcified lesions. In severe lesions, they've even been known to modulate into adipocytes, and we also see stem cell types derived from smooth muscle cells. So a really remarkable plasticity. And understanding the molecular basis of this plasticity will hopefully provide us with new approaches for prevention and therapy, where drugs can be applied to platforms such as balloons and stents and synthetic grafts. So there are many factors that influence smooth muscle phenotype under both normal and pathologic conditions. And these include the health of the endothelium and the diffusible factors it secretes, such as NO and prostacyclin, inputs from neurotransmitters that influence vascular tone, circulating factors, including platelet-derived growth factor, which is the main one we'll hear a lot about today, that initiates lesions in the intimal hyperplasia scenario, as well as other injuries, such as from excessive lipids, toxins, reactive oxygen, and inflammatory mediators that drive atherosclerosis and other lesions. And mechanical forces and extracellular matrix contacts have very strong effects on smooth muscle phenotype, and we'll talk a little bit about this at the end. So here's another example of this traumatic phenotype from a study in our lab. This is a mouse aorta where the contractile smooth muscle cells are in green. You see this nice elongated morphology, as opposed to where this neointima is forming in response to immune injury in a transplant vasculopathy model. And in cell culture, we can see these dramatic morphologic transitions as well. This is an example of the differentiated contractile phenotype with this elongated myocyte morphology. They express a repertoire of smooth muscle specific contractile proteins, also contractile phenotype specific calcium channels and transcription factors like myocardin. Whereas in the dedifferentiated or synthetic myofibroblast phenotype, they lose this myocyte morphology and take on a rhomboid fibroblast-like morphology. They lose their contractile proteins and express extracellular matrix proteins like collagens and fibronectin, different isoforms of contractile machinery, different isoforms of channels, and transcription factors like KLF4. This is a highly simplified schematic of the transcription factors that are at the core of regulation of this phenotypic switching. So in the differentiated state, CARG regulatory elements in the promoters of contractile genes bind serum response factor and myocardin, and these complexes drive expression of these genes. Whereas with a dedifferentiating stimuli like PDGF, the transcription factor KLF4 predominates and displaces SRF and myocardin and can recruit other inhibitory factors such as histone deacetylase 2. Now, our lab has studied drug-eluting stent agent rapamycin. This mTOR inhibitor is a highly effective drug that inhibits smooth muscle proliferation, but also promotes a healthy differentiated phenotype. This is a cartoon from the first-in-man trial where this very effectively inhibited restenosis in human coronary arteries. So we're very interested in the mechanisms of rapamycin action. And these images are actually from our initial study where we showed that in the dedifferentiated state in culture, the default is for smooth muscle cells in growth factors to assume this synthetic phenotype. These are actually cells that we treated with rapamycin and could induce this beautiful differentiated morphology. And this happens at the level of contractile protein transcription. So another major drug-eluting stent agent is paclitaxel that works by inhibiting microtubules to prevent proliferation and migration. The rapamycin family of drugs inhibit the protein, the mechanistic target of rapamycin, which is a master regulator of growth in the cells. It regulates growth and biosynthesis processes, including protein synthesis. Our lab has also shown that inhibiting mTOR with rapamycin promotes an array of transcription factors that promote this contractile gene transcription, also that inhibit cell cycle machinery. More recently, we found that the mTOR pathway also regulates an epigenetic regulator, TET2, that has profound effects on smooth muscle phenotype. And for the rest of this talk, I will focus on two areas that we covered in our review, epigenetics and the Hippo pathway, that I find extremely exciting for future therapeutic discoveries. So epigenetic regulation is an exciting mechanism because by changing the way the DNA is packaged, the way it's wrapped around these histones in silent inactive chromatin or in open and accessible chromatin, you can dramatically alter the whole program of gene expression in cells. And so this is achieved by post-translational modifications of the histone proteins around which DNA is wrapped, or modifications of the DNA itself, methylation of cytosines. So DNA that is methylated and deacetylated is compacted in silent chromatin, whereas open chromatin is demethylated and acetylated. So DNA methyltransferases have long been known to methylate DNA and promote this compacted silenced conformation, but the TET family of enzymes were recently shown to demethylate DNA. They oxidize this methyl group into 5-HMC, and this mark is characteristic of open and accessible active chromatin. We showed that TET2 is a master upstream epigenetic regulator of smooth muscle phenotype by regulating downstream transcription factors on both the contractile and synthetic sides. So we saw that injury and PDGF through mTOR signaling repress TET2, but mTOR inhibition with rapamycin activates TET2. When TET2 is active, it promotes this 5-HMC mark at promoters, including the promoters of myocardium and SRF, the master transcription factors for smooth muscle cells, as well as at the promoters of the contractile genes themselves. Now, on the other hand, TET2 activity promotes methylation and repression of the chromatin at synthetic gene promoters, including KLF4 and KLF5, which are master transcription factors for the dedifferentiated phenotype. Now, in addition to this catalytic activity in regulating DNA methylation, we found that TET2 also influenced histone methylation marks. And we speculate that this is through interaction with other chromatin modifying enzymes. We saw that TET2 activity was consistent with other histone marks of open chromatin at contractile promoters and repressed chromatin at synthetic promoters. And this is a subject of ongoing work in our lab by Raja Chakraborty to look at how TET2 intersects with other chromatin modifying machinery to regulate DNA accessibility in smooth muscle. This is a figure from our review summarizing the major epigenetic mechanisms that regulate intimal hyperplasia. And so, as we've shown, TET2 promotes this open, active state at contractile genes. We've more recently shown that ascorbic acid, or TET2, is a co-activator for TET2. And this allows us to enhance TET2 activity and help protect from transplant vasculopathy, this other model of intimal hyperplasia. Now, other kinds of epigenetic regulators, DNA methyltransferases and histone deacetylases, have been associated with the dedifferentiated phenotype. These repressed contractile genes in smooth muscle cells, and inhibitors of DNMTs or HDACs, these have been shown to inhibit intimal hyperplasia in mouse models. Now, there's a third type of epigenetic regulator called bromodomain proteins. These are a newer class of epigenetic regulators, but these proteins are called readers because they recognize histone modifications, including acetylation, and help promote the transcriptional activation of genes. And the protein BRD4 has been shown to drive proliferation genes in smooth muscle cells. And these various inhibitors of BRD4 have been shown to inhibit intimal hyperplasia in smooth muscle. So these epigenetic mechanisms are exciting new areas that are druggable. And one of the reasons I find this mechanism so interesting is that epigenetic regulation is intimately connected to cell metabolism. In this way, you can connect the metabolic and energetic state of the cell to the DNA accessibility and gene expression program, because so many of the cofactors and substrates for these epigenetic enzymes are direct metabolites of the TCA cycle or other important metabolic processes in cells. For example, acetyl-CoA is essential for histone acetylation, and alpha-ketoglutarate produced in the TCA cycle is essential for activity of TET enzymes and Jumanji, another epigenetic regulator family, whereas the methionine cycle generates the metabolites necessary for DNA methylation. So in this way, as we know, smooth muscle cells in the synthetic phenotype have very high energy demands, and by sensing the metabolic state through these intermediates, you can connect metabolism to the cell phenotype. So now, lastly, I want to talk about the idea of cell density and cell-cell contacts and contacts with the matrix as a way to influence cell state. So the mTOR pathway regulates nutrients and energy in cell growth. It also senses growth factors, but cell density and cell contacts are another critical input for cell growth and proliferation. And these pathways, these processes together influence cell fate decisions. The mTOR pathway, which we've talked about, which is targeted by rapamycin, regulates cell growth and cell size on an individual cell level. It is activated by growth factors like PDGF and senses nutrients, including amino acids, and energy like ATP versus AMP. So injury activates this pathway and growth factors. On the other hand, organ size is regulated. The ultimate size of the organ involves the size of the cells, but primarily the number of the cells. And cell number is sensed by the Hippo pathway. It's regulated by inputs like cell adhesion and cell density. And it gets its name Hippo because it affects organ size, and Drosophila has, cell organ size phenotypes that are pretty remarkable. And this pathway, the upstream inputs to Hippo that actually sense the density are not clearly defined at the molecular level at this point, but it activates a kinase cascade that represses these transcription factors, and these transcription factors drive proliferation. So it senses density, and these YAP and TEAD transcription factors have been shown by Jiliang Zhao, who I'm excited to see is here with us on this webinar today. He's really pioneered this field, and I was pleased to write this particular editorial on his paper. He's made important contributions showing that YAP and TEAD are required for intimal hyperplasia, and that there are connections between the mTOR and Hippo pathway whereby they amplify each other's activity. Here's another schematic of the Hippo-YAP pathway from our review article. And as I mentioned, the upstream most inputs aren't directly known, but we do know that G protein coupled receptors can influence the activity of this pathway in different ways, including the prostacyclin and thromboxane receptors, which are known to regulate smooth muscle contractility and phenotype. So the prostacyclin receptor inhibits YAP signaling, where the thromboxane receptor promotes it. So agonists or antagonists of these receptors are a promising way by which we can modulate Hippo-YAP activity. And mithromycin A is an SP1 inhibitor that's been shown to influence expression of YAP and TAZ as another way to modulate this pathway and prevent intimal hyperplasia. So yet another schematic of the Hippo pathway. I wanted to include this because it shows some of these upstream inputs, such as integrins, mechanical stiffness and force, G protein coupled receptors and their ligands, and cell polarity regulators, all of which can feed into this kinase cascade that ultimately regulates whether YAP, TAZ are localized to the nucleus, where they can cooperate with TEAD to drive pro-proliferation transcription factors. So this is from the same review article, just showing the many ways, mostly indirect ways, but some direct ways by which we can manipulate the activity of this pathway, which could have important implications for modulating smooth muscle phenotype. So we'll hear more about these pathways. From Dr. Miyachi, we'll hear about imatinib, which is an inhibitor of the PDGF receptor. From From Dr. Smith, we'll hear about a cation channel that influences contractility that reinforces a smooth muscle phenotype. And from Dr. Atkins, we'll hear about some matrix remodeling mechanisms that will influence cell-cell contacts and cell phenotype. So I'm very much looking forward to these next talks. And I just want to thank and acknowledge all of my co-authors on this JVS VS review who are highlighted here in red. Alan, who invited us to write this article, and all of my collaborators and lab members who've made our work in this field possible over the years. So thank you for your attention.
Thank you, Kathleen. That's a very comprehensive and interesting talk, and it certainly points to the future, I think. Any questions now for Kathleen, you can post it in the chat, and I promise that Kathleen will answer you in the chat. Alan has already posted one.